All right, we can start to get this webinar started. I'm Rachel Hostetler. I'm a scientist at the Allen Institute for Brain Science, and today I'll be presenting on the data challenge results of our data challenge called MapMySections. And with this webinar, we'll be having two teams featured to present their winning algorithms. And the first team consists of Daniel, Austin, and Boris from Stanford University, and our second team features Nikhil from the American Physical Society and Chaitali from the NIH. And as a quick reminder, this webinar is being recorded, so it will go live on our YouTube channel after this webinar. And the agenda follows that we'll have just a brief introduction to the data challenge, the purpose of the challenge. Then team one will present for about 20 minutes, plus we'll have some time at the end for some Q&A. Then team two will present their results. Then we'll have some more time for team two to have Q&A as well. And if you'd like to learn more about this data challenge, you can go to alleninstitute.org/events/mapmysections. And we do also want to acknowledge our funding for this particular data challenge. All right, so the goal of the challenge was to make a connection between the data that we have in the Allen Institute Genetic Tools Atlas with the data, specifically the spatial transcriptomic data that we have in the Allen Brain Cell Atlas. So these are two different atlases featuring very different types of data. And the goal of the challenge was to make connections between these different data sets. And so the plan was to take the data from the Genetic Tools Atlas, which features serial two-photon tomography images labeled in brain sections from different AAVs or labeled in different genetic ways, and then try to map that onto the spatial transcriptomic data that's featured in the Allen Brain Cell Atlas. And the motivation behind this data challenge was we know that the Allen Brain Cell Atlas is a useful tool for looking at different cell types in the brain. But then if someone wants to use that information to design an experiment, they're probably going to next ask, is there a genetic tool available to label this particular cell type that I'm interested in? And so the goal of this challenge, again, was to link these different data types together. And there were two different judging categories for this data challenge. The first category was purely just based on accuracy and how accurate the mapping was. And team one, featuring Daniel, Austin, and Boris, were our winners for the algorithm accuracy. Their tool is called Unravel, and they'll be presenting first. And then our second team, our second division for judging was based on how user-friendly was this tool, how easy was it for the judges to access and understand. And the winners for this category were Nikhil and Chaitali. And their tool is called Genetic Tool to Cell Type Bayesian Mapper, and they'll be presenting second in this webinar. All right, and I'll stop sharing my screen and I'll allow the first team to begin.
All right, so I'm Dan, and I worked with this on this with Boris and Austin, and we developed Unravel, which is a Python package and command line tool that we've used to automate the analysis of 3D images of mouse brains, either from light sheet fluorescent microscopy with cleared brains or with serial two-photon tomography. And we use this to generate maps of the labeled cells. So our most common use case is to immuno-label the brains and for CFOS, so we can look at as a proxy of neuronal activity and clear them, and then use a light sheet microscope. But we can also use the same tool for looking at the distribution of AAV labeling, transgenic lines, etc. We can perform either region-wise analyses or voxel-wise analyses, looking at label cell densities within regions of interest, or label densities like axonal innervation. Then once we've identified hotspots, we can look at what Allen Brain Cell Atlas cell types are present, as well as explore gene expression from the Allen Brain Cell Atlas. And then we've also developed ways to visualize the data, either in 3D or to um, provide a detailed characterization of the anatomy of a hotspot or the cell type proportions. And so the overall goal, as Rachel mentioned, is to basically take 3D images of mouse brains and to identify the top cell types. and then use that to guide tool selection, whether that's antibodies, enhancer AAVs, or transgenic lines. And our approach can take a variety of supports a variety of data input types. So either if people already know a region of interest, they can pretty easily analyze that. We can explore data from the genetic tools atlas, like as in this challenge. Or if scientists have their own experimental data from cleared brains or serial two-photon tomography, that can be analyzed. Or if they have statistical maps like p-value maps, that also works. And so I'll talk about some background on using Unravel for brain-wide mapping of fluorescent labeling, show an example of integrating maps with the Allen Brain Cell Atlas data, and then go into our approach for MapMySections. So the 3D images of mouse brains from either of these modalities are really useful for comprehensively characterizing the labeling of cells or proteins. For example, we can look at biomarkers or lesions in disease models or better understand genetic tools. And so we initially developed Unravel as a way to analyze light sheet image, automate analysis of light sheet images. For example, we've characterized the distribution of CFOS induction from psilocybin treatment, which is a psychedelic drug. And we can see that there's a lot of CFOS expression in the cortex, but some in subcortical regions as well. And we use these sunburst plots quite a lot, both for looking at regional composition as well as the cell type composition. And so the wider the slice of a pie, of the pie, that indicates more volume. So you can see that there's a lot of the volume is from the cortex. And then inner rings represent coarser levels of granularity, like cortex versus subcortical nuclei, and outer rings represent finer granularity, like specific subregions or cortical layers and so on. And our code is publicly available on GitHub via this link, and there's a link there for our documentation as well. And we've automated analysis of these types of images. Our tool has kind of modules for registering images, warping images to and from atlas space or between or to tissue space, segmentation, quantifying cell types that are present, and visualizing the data. And so we mostly focus on voxel-wise analyses because that really allows us to hone in on the areas where there's the greatest difference in signal, and then provide this regional breakdown in a variety of formats. And then lately we've been integrating these maps with data and tools from the Allen Institute that have been really useful for deepening our insight into this data. So for example, after we found an interesting hotspot where CFOS was induced in the central amygdala and basolateral amygdala, we wanted to look at what cell types were present within that hotspot. So we could take the spatial transcriptomics data, the MERFISH data set from the Allen Institute, and basically mask out and drop any of the cells that are outside of this hotspot, and then look at what cell types were within the hotspot at the neurotransmitter level, the class level, subclass level, and so on. And then we can also quantify the proportions of cell types that are present. So for example, we're looking at glutamatergic cell types on the left and GABAergic cell types on the right. And we can look at the proportion at the class level, subclass, supertype, or cluster level. We can also, once we find a cell type that is enriched in a region of interest, then we can try to identify markers that are relevant for labeling or manipulating that population of neurons. So for example, we looked at the enrichment of gene expression within this hotspot relative to the rest of the brain, and looked at all looped through all the possible genes and identified one that is a really good match for this. And then we can confirm that there's decent cell type specificity of this marker gene expression using the single cell RNA-seq data, also from the Allen Institute. So for Unravel, we have a website indicated above that has detailed documentation both for mapping fluorescence throughout the brains and for the MapMySections challenge. So there's a page on installation. There's an analysis guide for the light sheet and serial two-photon data, as well as video tutorials. Each script has a detailed help guide that's accessible either online or via the command line. You can write the command and then -H to view the help for the script, and we've formatted that in an easy to digest way. And we provide hints on, what is the next command so people can, work their way through the process without necessarily having to go back to the online documentation. Or you can, we have ways of discovering commands like listing out common commands or filtering, or organize the command such that you can type the prefix like MMS for MapMySections and hit tab to look at, what other commands might be relevant. We also have a page that goes over our approach for MapMySections in detail that goes through all of the commands that we ran for this challenge. And we'll record a video tutorial and post a link for that on this documentation. So then for our general workflow for mapping fluorescent labeling is to take the autofluorescence image because that better matches the average template image and resample that to 50 micron resolution. We clean up the brain and then we register the average template with the autofluorescence. And then we can use transformation fields from that to warp it into standard CCF atlas space. We use 30 micron atlas space for our analyses generally. And then for mapping labeling of fluorescence, you typically use rolling ball background subtraction to remove autofluorescence from the channel that has more labeling. And then we can warp that to atlas space for all of the samples where we can perform voxelwise analyses and generate p-value maps that you can see in warm colors. And we use FDR to correct for multiple to define clusters of significant voxels as shown in white. And then we can take those hotspots and warp them back into tissue space and use elastic to segment the CFOS positive cells in this case to look at things like the density of cells or the density of axons or whatever else. And we use this as a way to validate that these hotspots are really due to a biological difference that we care about as opposed to just, the antibody aggregating or something like that. We also developed a website in collaboration with Metacell to deposit our data. It's interactive, so people can then explore the statistical maps that we've generated and published on. And so now I'll transition into our approach for the Map My Data Challenge. So, if you identify a hotspot in the brain, how do you use the spatial transcriptomics data to basically characterize what cell types are present there, and then create kind of spit out what the proportions of each cell type that are present within that region. So there are a variety of challenges that we needed to solve to address this. One is we wanted to detect only the cells and not diffuse axonal innervation, so that we could better link the cells to the merfish cells. And then we also wanted to distinguish neurons versus non-neuronal types. So for these steps, we downloaded the training and test data directly from the genetics tool atlas. They're multi-resolution XAR images, and we just downloaded a resolution that worked well for showing morphology. And we converted the channels into separate TIFF series for easier processing, and organized the samples automatically and can crop them automatically too, to save disk space and speed up processing. And then we can segment the cell bodies either for neurons or oligodendrocytes. We can segment astrocytes and also endothelial cells using elastic. And then we perform for spatially filtering the merfish data to look at what cell types are present within the merfish data. We have to perform registration. So we take the autofluorescence channel from the image and we register it with the average template. And then we can use that to warp the segmentations into atlas space. Then to have more complete or better coverage, we take those segmentation images, we mirror them, kind of flip them, and then we combine that with the flipped version with the original version. And then we warp those combined versions to merfish space and use the cell segmentation as a mask to spatially filter the merfish cells. So then as a final step, we try to make the data digestible, so quantifying and visualizing what the cell types proportions that are present across all levels of the hierarchy, and as well as for specific brain regions or for the brain as a whole. So this is the Allen's Institute's Genetic Tool Atlas website. So you can go there and filter the data by modality to look at serial two-photon data. And then you can filter it further to find brains of interest, like if you're interested in a certain cell type or region, and download metadata associated with that can be used as an input for our scripts for automatically downloading the ZAR images. The ZAR images have multiple resolutions. If you download the whole thing, they can be 200 to 500 gigabytes. So we wrote code to only download a specific resolution. And then once, to segment those images, we used Elastic, which uses sparse user input and kind of an older machine learning algorithm to segment the cells. So for example, you can paint on the cells or you can paint on the background, and then that you can basically iteratively teach Elastic to segment, labels of interest or features of interest. And so from the nearly 400 brains from the MapMySection challenge, they had some ground truth data from FACS cell sorting and single cell RNA-seq. And we selected a subset of them, 25, that had high cell type specificity for different primary visual cortex cell types for training at the Elastic project. And we've made this project publicly available and have a link to it in our documentation. And so we used 16 neuronal brains and 3 brains each for oligodendrocytes, endothelial cells, and astrocytes. And so for neurons, you can see that we segmented the cell bodies. And for oligodendrocytes, we also segmented the cell bodies because Elastic wasn't able to distinguish them morphologically from neurons. And we focused this is showing the corpus callosum, but we actually focused on the anterior commissure. And basically, we looked at the prevalence of the oligodendrocyte segmentation in the anterior commissure, and if it reached a certain criterion, then we made the assumption that it was a sample that had oligodendrocytes. For endothelial cells, we could segment those as well. And if the majority of voxels were segmented were endothelial cells, then we assume that those samples were primarily endothelial. And the same thing is true for astrocytes. Then we needed to warp those segmentations into atlas space. So for this, we took the autofluorescence channel, or whichever channel had less fluorescent signal, and we registered that with a serial two-photon template, and then we could warp the samples into atlas space. And once the segmentations were in atlas space, we decided to try to improve this with the merfish data set. There are some missing sections and there's some portions of the tissue that are missing. And so, in the case where the brain has sparse labeling of cells, we wanted to make sure that those overlapped with merfish sections that were present. And also, we just wanted to fill in the data set more completely, or where the cell bodies are located more completely, so that we'd have more cells to quantify in the merfish data set. And so for this, we took the original image, we flipped it, and then combined the original and the flipped image and used that for warping to merfish space. And to warp to merfish space, we took our average template and also the average template from merfish space, and we registered them together. And then we used transforms for that for warping our data into merfish space. And so you can see in the background is the CCF template from the original CCF template that we used that was warped into Merfish space. So the registration and warping is good. And so for, after we identify regions of interest or hotspots, we use this cell metadata CSV file from the Allen Institute to basically look at what cells are present. So each row in that CSV represents a cell, and it's richly annotated. And as per their documentation, we add to it a bunch of other columns to more, to do things like filter by region. So it's relatively trivial to filter the data by region, but it's more complicated to develop a way to spatially filter the data using a mask. So I'll show an example of each. So this is for filtering by region. We can basically take the Merfish data and we filtered it to only include cells from the nucleus accumbens. And then we can quantify the proportions of cell types across in the nucleus accumbens at each level of the cell type hierarchy. So you can see D1 and D2 medium spiny neurons as you'd expect. And we can also look at gene expression for all of those cell types. And so we can either do this using the imputed data or the data that was directly measured. And you can see that this works well for picking up, DRD1 and D1 medium spiny neurons or DRD2 and D2 medium spiny neurons. And so for spatially filtering the Merfish data, as an example, I'm showing the periaqueductal gray matter in white as a mask, and use that mask to filter out Merfish cells where the coordinates were outside of that mask. And then I'm plotting in green, the Merfish cells that were present within the mask that fell within it, and then quantifying that on the right. And we can see that the vast majority of cells that remained after filtering were correctly in the periaqueductal gray, and the remaining cells are at the boundary. And so then this is an example from one of the images from the MapMySections challenge, where we have the segmented cells in atlas space and colored it according to Allen Institute standards. And then, this is just a coronal view of that and the regional composition of the labeled cells. This is an example of that brain with the fluorescent signal, and you can see, strong labeling in this particular cortical layer, and you can see the neuronal stomata that we've segmented. And then after this spatial filtering approach, you can see here what cells were present and remained present in the Merfish data set, either showing the structure or the neurotransmitter class or subclass. And we could quantify the cell type proportions across the brain as a whole, or for specific regions, like for the challenge, we focused on the primary visual cortex. And so then for future directions, we want to optimize further our MapMySection strategy. So one of the limitations with our approach is that when there are cell types with overlapping distributions, like D1 versus D2 medium spiny neurons are intermingled within the striatum. And so we want to be able to distinguish, them using we think we can use their known projection patterns to basically distinguish the cell types further and to basically filter with an additional step. And so for example, for D1 medium spiny neurons, they project to the globus pallidus internal, or the substantia nigra reticulata, whereas D2 MSNs project strongly to the external part of the globus pallidus. And so we can look at the fluorescent signal within those known target regions as a way to then refine the proportions further. It's a little bit more challenging for sparse interneuron types, but there may be ways to address this as well, just like by looking at cell density. And we also want to expand our integration with the Allen Institute tooling. So for example, after you find a cell type that is prevalent within a region of interest, we want to make it easier for people to look up relevant promoters or enhancer AAVs that could be used for their either labeling the cells or manipulating the cells experimentally. And then finally, it would be useful to develop a graphic user interface. So this is all we mainly use the command line, although you can use Python, or Unravel as a Python library, but we would like to make the tooling more accessible in general and develop a graphic user interface in the future. So with that, I'm happy to take Austin and Boris and I are happy to take any questions.
All right, thank you for that presentation. One question that we can start with, just a general question regarding the data challenge. Through all the different steps that you did, what was the most challenging aspect of it?
I mean, I struggled at first with just, downloading the ZAR images from the genetic tools atlas that, and figuring out, okay, these are huge and downloading a specific subset. That was one challenge. Figuring out how to do the spatial filtering and the code for that was challenging. Um, Other aspects of it, we had been developing Unravel to basically take advantage of the Allen Brainsol Atlas data already. So we had in place, the ability to create those sunburst plots and show the data in some of the ways that were shown. So it was fortuitous that, this challenge came out, but, those were our biggest challenges.
Another question regarding the challenge. So part of this challenge was not only to solve this problem, but also try to make something that's user-friendly and accessible to other people. So keeping that in mind, and in the spirit of open science and sharing everything, did that also affect how you approach this challenge, as opposed to just creating a tool that's, just for your lab to use?
Yeah, we tried to make it fairly general. So we wrote code that makes it easier for people to mine the data from the Genetic Tools Atlas and not just using the data from this challenge specifically. And um, our tool set up so that it can process data from, either light sheet data or serial two-photon data. So there's some flexibility in terms of, image formats. It can work with Zeiss CZI images, ZAR images, HDF5 images, TIFFs, etc. And we try to make it fairly agnostic to the uh, image types. And then we try to keep in mind as we're writing code what functions may be generally useful. And so we, kind of create functions that people can import into scripts in a modular way. And, we've developed some, put together some documentation to hopefully make it easy for people to follow through the process. Of course, if they don't have any experience with the command line or with that may make it a little bit harder to use, but we have some links that people can use to get oriented to that. And as I think personally, the command line is a wonderful tool because it just opens up so many possibilities. If you want to process, batch process any files like renaming files, it's super easy for doing that. So learning how to write a for loop, learning about like working directories and paths, file paths, I think that those are really fundamental concepts that people can benefit from learning. So, in the future, we'll try to make the tool more user friendly. I know not everybody wants to learn that kind of stuff. So, hopefully in the future we'll develop a graphic user interface that people can use. But I think we need to optimize our approach a little bit first before we put the time into, having kind of a concrete tool in place for that.
Thank you. And if there's any other questions, attendees can put their questions in the question box. And with that, I'll ask a final question. Were there any particular cell types that were especially difficult to work with in this challenge, whether that be, large classes like glial cells or like, particular types of neurons?
Yeah, it was easier. Most of the brains were neuronal from the challenge, but there were some that had astrocytes, oligodendrocytes, endothelial cells. Those were more challenging. Elastic is really easy to train and work with, so we went with that. But we might have better performance of classifying cell types based on morphology using a convolutional neural network. For example, a UNet architecture is pretty commonly used for image segmentation. And that might be able to factor in, like with Elastic, it almost has kind of tunnel vision. So it works well for adapting for local changes in brightness and kind of looking at, a very focal pattern, but it doesn't, it's harder. It's not, it doesn't work for looking at if there is a dendrite or an axon that's strongly labeled, it might mistake that as an endothelial cell, for example. So, certainly not perfect. So I think, using a variety of approaches is good, and we should certainly explore deep learning methods in the future.
All right, thank you for those answers, Daniel. And with that, we can move on to the next presentation from our second team, Nikhil and Chaitali. Oh, Nikhil, I believe you're muted.
Sorry. Sorry about that. Can you see my screen?
Yep, everything looks great.
Okay. Sorry about that delay. Yeah, I would like to first thank Rachel and the organizers of this challenge for selecting our tool as one of the winners and allowing us to give a talk here. So this is a tool. So to introduce myself, I'm Nikhil. I'm a computational physicist and an editor at American Physical Society. And I developed this tool with Chaitali Khan, who's a molecular biologist at NIH. So we developed this tool called a generic tool to cell-type mine Bayesian mapper, which we call the GT to CT. Why we call the Bayesian approach will become clearer in a bit. So for those of you following this talk, you could scan this QR code and it'll take you to the GitHub page, and you just have to open the notebook there, and you can follow along and scroll and see what it does. So as you can see, we have kind of designed the notebook so that it's self-explanatory in nature. So I'll spend most of my talk trying to explain the underlying approach. And the last part of the talk, I'll quickly walk you through the notebook and as a user, what kind of variables you need to change and what you need to focus on. So with that, let me give a basic mathematical idea behind the tool. So to recap, the challenge was the following for the MapMySection challenge. So the idea was you're given the GFP or the RFP positive cells, which are targeted by the enhancer AAVs or the transgenic mouse lines. And you want to map fractions of those cells, which I denote by these green crosses, to their transcriptomic cell types. So the extra information that we are actually given is the spatial distribution of the transcriptomic cell types, which are encoded in these mesh cells, which are available in the Allen Brain Cell Atlas and published here. So the idea is to basically map the things on the left, the GFP cells. They could be GFP, RP. I'm just going to call them GFP in the stock. So from the GFP cells to the things on the right, to the mesh cells, for example, what fraction of them could be belonging to the subclass level L23, or what fraction of them are L5, and so on. And also the challenge was, it could be in general, but here in this talk, I'm going to focus on the primary visual cortex, which is the WISP. So this is the challenge. So the basic overall idea of our approach is the following. So we want to assign to each cell, which I'm going to assume for the first part of the talk to be point-like objects. Physicists take all kinds of approximations like spherical curve approximation. So in this talk, I'm going to take the approximation that a cell here is a point-like for this first part. So it's going to be a cross, and we want to assign to each cell the probability that that particular cell belongs to a cell type, which I call C, which, for example, this cell at that location in the CCF coordinate is of type C1, which I would call the probability condition probability on the position. And this cell is of the type C2, conditioned on its location, and so on at different locations. So once you have the probabilities for the multi-cell types, then the fractions are basically obtained from these probabilities by averaging over the space or marginalizing over the volume of WISP. So that is the underlying idea. So we are not given the probability distributions. The data is really the MERFISH data set. So we want to learn these probability distributions conditioned on the locations from the MERFISH data. For this, we use a very simple neural network, which is the multi-layer perceptron, which is a fully connected layer. We use about 5 layers with 64 hidden units in each of those layers. So the input to the multi-layer perceptron are the CCF coordinates scaled to make it stable. And the output are the logarithm of the probabilities for the 28 subclasses in WISP, or for different regions, it would be the number of subclasses or the cell types in that particular brain region. So the way we train this data is quite straightforward. The data in this case for training is the MERFISH data in the WISP areas. Here I've shown a Z slice of the brain in the WISP area. So it's basically a three-dimensional region in general. So it's a three-dimensional probability distribution, even though I've shown it in 2-dimension here. So each of them is a MERFISH cell of different cell type. So we divide. So there are about 60,000 cells in WISP, but we kind of enlarge the data set to about 200,000 by looking at WISP and also some surrounding region in the WISP. And the idea behind the training is that we kind of randomly pick a MERFISH cell from WISP. Its location and the cell type are known, and we feed in this location into the MLP and predict its cell type, and then use the stochastic gradient descent to slowly increase the probability of predicting the correct cell type. And repeat this process by picking another MERFISH cell and so on. And then we have the test data set, 10% of the data, and see how it converges to the correct number. So we were able to see, like, with the top 3 probability were around 76% probability success rate with this approach. So with this, we now have the setup to predict the 3-dimensional multi-cell probability distribution over the 3D WISP. With that, we have one more extra information for which we knew we want to use from the beginning of the challenge. It is that the cell type specificity of the genetic tool. So they are not genetic tools which kind of flare up all parts of the cells, but they are enhancer genetic AAV genetic tools which target very specific cell types. That's the way the alleninstitute has designed it. So this plot here, which I've taken from Ben Simon et al., the recent Cell paper, it shows the genetic tools on this y-axis. On the x-axis is basically the WISP subclasses, and the size of these circles here say what fraction of those subclass cells belong to that subclass got targeted by that enhancer AAV. So as you can see, for each, if you look along some kind of given a specific enhancer, you can see that only like maybe 1 or 2 have a substantial fraction, maximum 4, and then like very less number of fractions are getting targeted by a tool. So cell type specificity is now included as a part of G2 to CT as an implicit bias. So we wanted to include that as a constraint in the algorithm. So for the demonstration purpose, let's take for a moment that the genetic tool only targets like 2 cell types out of the 28 subclasses in WISP. For example, it could be cell type C1, C2, which I call the subclass K1, or it could be C1, C3, which I call K2, and so on. There are multiple combinations of these 2 cell types which could be picked out of it. The constraint is that the cell types which are getting tagged by the gene tool should be the same at all locations. Either it picks cells from a given K1 or a given K2, but that's the only subset from which they can be picked. So the idea behind now choosing which of these is K1 should I pick is so that the best set of optimal set K star should be the one which maximizes the probability of generating this GFP data. So it's basically the probability that, given this hypothesis that cells are being picked from K star, what's the probability that the GFP data, which is given to us, is maximal? It's a maximum likelihood method. So this probability of the data factorizes into is just a product of that the cell at X1 belongs to one of the cell type in the subset Ki, times the probability that that a cell at X2 is also picked from the same set Ki, and so on and so forth. The probability multiplies. So we want to maximize this product of probabilities. If you take the logarithm, it becomes the sum of logs. And you want to basically choose the K, the subset K, which maximizes this thing. And this probability, which is the probability to find a cell being picked from Ki, can be related to the MLP that we trained via the Bayes theorem. So we already have generated that, tuned that MLP, from which we can get the subset K star. So once we have the ideal set K star, you can figure out the fractions by just marginalizing these probabilities over Wisp. So this term here is basically the full probability that we learned using the MLP and projected onto the subset ideal set K star. So from 28 subclasses, we have reduced it into, we kind of put a constraint that it's now part of maximum K cell types of 4, for example. So in this challenge, we know that it's kind of specific, but we don't know the value of the size of the subset K star, which I call the K again, not a scripted one. And so we repeated this analysis with multiple values of the cell type specificity K from 1 to 4 and changed it. And we took the model average of this to predict the cell type. So this is the underlying idea. So the point now, until now, kind of for simplicity, I kind of assumed that the cells are all point-like, which is not true, as we saw from the last talk, where great care was taken into account for taking into the size of the neuron and so on. For our challenge, it was even more higher level complication. It's that the STPD data that was given to us was at a very low resolution, namely at 25 micron voxels. So these kind of ideal image I showed before is no longer true. It kind of blocks up into these kind of voxels and kind of brightness, which are over 25 microns. So we had to work with that. So we had to start from the scratch. We didn't have the time to do registration and all that. So we had to work with what was given. It was 25 micron voxel. So we gave up on certainty at that level. And what we kind of began to do is to treat the GFP cell also probabilistically. So basically, we just wanted to ask, what's the probability that you find a GFP cell in this particular voxel? So for that, we kind of, in this example, this is also probabilistic in some sense because you have probability 1 at all these sites, which are kind of represented here. Now we need to generalize into kind of a smoother probability over the 3D Wisp. And we decided to assign a GFP probability being proportional to a thresholded voxel intensity. If there's time, I'll come to that point a bit later. So once we have this probabilistic approach to the GFP, we also have to generalize all the expressions I showed before. For example, instead of maximizing the probability of maximum likelihood approach, we had to minimize the cross entropy loss between the probability, cell type probability, and the spatial probability of GFP, which is given here. Even though they are different terminologies, the resulting expressions are pretty similar, except in a GFP probability here. So we want to maximize this to get the best set K star of cell types. From that best cell K star, we can find the fractions by summing this expression over the full Wisp, not just over GFP. And so that unweighted by their GFP probabilities. So this is the idea from which we obtain the results. So quickly, I'll show some results from the MapMySections data set, whose actual answers were announced after the challenge was over. So let me take 3 broad classes. First is, for example, the glutamatergic cells, which are easier to, in general, to identify even by the eye because they are pretty localized to layers in the brain. So this is, for example, a test case number 1 in the data set if you have access to it. So this is the AAV enhancer targeted towards the L4-IT cell type. So here on the left, I show you the prediction from the GTT-CT with a cell type specificity of K equals 4 baked in. And these are the predictions for the fractions for different cell types. And on the right, I show you the actual fractions here. So as you can see, as expected, because it's easy to predict these cell types, it gets the top one pretty close. And but even though it misses the L6, at least that cell, it gets the fractions of cells which are actually present pretty well. And on the bottom, I show you the visualization of what the prediction is. The gray ones are the Medfish cells in the Wisp in a specific Z slice. On the green, I show you the probability density of multi-cell type in these 4 cell types that have been chosen. And on the red is the GFP distribution that has been constructed. So it kind of tells you that it's just a sanity check that why you got that answer that you got, and it tells you why you got this answer. And more difficult than the glutamatergic cells or the GABAergic cells, which are not that localized and diffuse and less brighter. And so here's an example from here, data set number 7. which is targeted toward the SST GABA cell types. And here, the actual set, you can see that it's targeting almost 100% of the SST GABA cells. So when we start with our G2CT with a cell type specificity of 1, it's getting the right cell type right on, even with this approach. But the moment you kind of put an uncertainty about the cell types specificity, it tries to struggle a bit and kind of tries to overfit the data, including multiple dominant cell types. But at least it kinds of getting confused with the GABAergic cell types rather than glutamergic. So, but at the end, it does a decent job. And here again, I show you a particular representation of this for this particular Z slice. And here is the non-neuronal cell type, which is targeted towards endo, which was a pretty difficult thing from the beginning. So this was kind of the test case where both me and Chaitali knew that we were doing something right, kind of a eureka moment, the moment we were able to predict, at least get those cell types correctly. So here is a example here where it's able to predict the endo cell type, even though the fractions are kind of off from the actual result, which is 80% here. And as you can see, it's kind of diffuse and predicting it. So I don't want to leave you with the thing that, oh, it's kind of not performing well. So I want to, again, flash a cell type from the glutamergic cell that, yeah, it's a nice try to work with. So let's leave here with some optimism. So here's one more glutamergic cell where it performs pretty good. So in the remaining part of the talk, I'll just quickly walk through the notebook and see what you need to focus on as a user. The notebook, so the way to install is the instructions to install the thing are given at the GitHub page. So when you execute those statements there, which are shown in red, you should see something like this, a lot of installations. And once you activate the virtual environment, you can open it using the Jupyter notebook here, which should pop it open in your web browser like this. And then you can keep executing them cell by cell. And there is enough, I hope we have provided enough instructions on top of them for you to understand what's happening. So the notebook basically has 4 broad parts to it. So I'll just quickly go through them. The first part is where all the data from the Allen Brain Cell Atlas are being loaded. For example, there's brain annotation, the coordinate system information, and the MERFISH data, which is reflected as was done in the last talk and projected onto the WISP area or whatever area that you want to target on. That can be, so as a user, the place where you want to focus on is this variable where you can specify what area you want to concentrate on. And one more thing it does is for this challenge, so these are 25 micron voxelated images, whereas the MERFISH is in 10 by 10 by 200 micron voxels. So to avoid any kind of error in learning using this via interpolation errors, so we chose only those slices from STPT, so that the closest distance to a neighboring MERFISH is within a certain tolerance that you can set as a user. So in that way, we kind of lost certain slices, but because we are interested in fractions, as long as we are sampling them, we should be able to get it right. And the second part of the notebook is where you either load or train a new neural network to learn the 3D multi-cell probabilities. So the cell that you want to focus on is here. For example, here we are setting the probability model to be the multi-layered perceptron. If you're using for WISP and just want to use the one we have used, you just have to load it in. Or you can even bring your own model as long as the input is some scaled X coordinate and the output are the number of subclasses or the cell type level, hierarchical level that you're interested in. So if you set this variable to true, it will just load the pre-trained model here whose weights are given in this directory, and it will not deactivate all the cells where you have to train. Or if you set it to false, then it will activate all the cells where it involves the training and it will start training the model. This is all part of the same notebook, so you don't need to write a new code for that. But it's straightforward PyTorch code. And the third part is where you as the user will be uploading your image, the serial two-photon tomographic image as the input. So you want to place your nifty.gz image in this directory. For example, the red and green named in this way channels. At present, it doesn't have the capability to choose which channel you want to look in. It expects you as a user to specify the correct channel to look at. And once you select the right channel, the rest of the cells will compute a threshold voxel intensity based on slice-wise. And then from the slice-wise, it will compute a global threshold and place the global threshold on the full WISP slices to avoid any bias on a cell by slice level. So for example, here's the unthresholded WISP slice, and here's a globally thresholded image. And once you have done that, it will compute the GFP probabilities based on the thresholded voxel intensity said to be proportional. Yeah, so that is the model that we use for GFP probabilities. And part 4 is where it puts together the GFP probability densities and the multi-cell type probability densities that we learned together to predict the cell type fractions. So the user has to focus on this variable here, k-target, which is the cell type specificity. For example, it's said to be 3. And that's either you have a knowledge of the cell type specificity for the gene tool that you work on because you know what it is, or if you don't know, you set your right prior that you want to put in or vary it to see the sensitivity of the results to the cell type specificity information, and then do model averaging of the choice you want to do, maybe better than what we have done. And using that, it will compute go through all this cross entropy minimization, choosing the correct optimal subclass and so on. And then in this cell type, it will output the fraction of cell types here, which should sum to 1. And these are the cell types and the fractions. And as I've shown before, the last cell in the notebook will output the visualization of the information. Namely, these are the Medfish cells are shown for visual aid purposes, and the green one for these density plots are the multi-cell probabilities spatial distribution for the 3, in this case, cell types. And on the red is the GFP voxels based on the voxel intensities, thresholded voxel intensities. So this completes the cell. And just about the future, what you can improve. For example, as I told you, we have been using the 25 voxel images, and the tool expects the user to place that, do the registration to CCF and place it in as an input. But over the challenge, we learned that it can be registered and so on. But for example, it should be straightforward for the experts to kind of include that pipeline as part of the tool, and maybe even register at resolutions which are 25 micron, maybe 10 micron, which should definitely improve the accuracy of the tool. Is MLP, the multilayer perceptron, the best architecture for learning the multi-cell probabilities? We don't know. It works. It kind of makes the approach mathematically kosher. But one could think of generalizing the conditioning the cell types on X CCF locations, kind of generalize it to a conditioning of the cell types on the neighbors of C. For example, once you do that, all kinds of neural network architecture comes in, like graph neural network, attention mechanism, and so on, something for the future. And thirdly, is the most, I would say it's an important thing to improve on, is that we have set the GFP probabilities being linearly related to the thresholded voxel intensities. Nobody told us it has to be so. It's kind of a ad hoc approximation on our part. So this, instead of doing that, what really should be done to make it more rigorous is to learn the nonlinear relationship between the GFP probabilities to the thresholded voxel intensities. For example, one way to look at it is shown here. So it's basically look at this patch here and then kind of learn this linear relationship to the probabilities via some neural network of your choice, and learn this neural network weights. For example, you feed in this into the GTCT tool, predict the fractions, and using some test data, tune the learn to increase the correctness of the fractions. So for example, there are about 300 cells. Training data was given as part of the challenge. We did not really use that in a very quantitative fraction, which is a shame. But in this approach, we should be able to like quantitatively use that training data also to improve the model, which we did not. So this is definitely a place for improvement. Yeah, that's it. That might take both me and Chaitali can take questions, etc.
All right, thank you for that presentation. One question that we have is, this question comes actually from a fellow physicist regarding your selection of cell type set K. can you get any utility out of considering similarity or differentiation between cell types, i.e., are there combinations of cell types that can be ruled out because no reasonable genetic tool would target them, perhaps?
Yeah, this is a good question. So at the initial part of the challenge, both me and Chaitali were thinking about it. Like, for example, at the moment, I'm assuming that all the all the combination of cell types to be the same, and we are choosing them. And we haven't really used any biological fact about these combinations being targeted by an AAV enhancer as such. Maybe there must be some way of using the single-seq data for each of those combinations and the relation to the genetics tool to kind of place a prior distribution on the cell type combinations themselves. It was just getting too much for to think about it or implement how to do that. So we kind of put a uniform prior over all combinations. So, but you're right. I mean, there must be some way to use the biological information on placing a prior or rule out, place more weight on certain combinations than certain combinations. Yeah, I don't know, but I suspect it should be possible.
Thank you. And another question we have is, you might have briefly answered this at the very end of your presentation, but how do you know which K to choose in your examples when different Ks give very different results for mapping?
So that's why we know it's from the Ben Salmon et al. Cell paper. We know it's kind of targeting less than 4. Of course, it's not 0 for the rest of the thing. It's kind of maybe 1%, but the substantial fractions, let's say about 10% or so, is mainly on few of the less than 4. So we think so we decided to put 4. Of course, there's no reason to say it's not 1 or 2 or 3. So that's why we chose all 1, 2, 3, and 4 and kind of model averaged it. So that's what we did. And in fact, from the Ben Salmon et al. Paper, there's extra information, which is the histogram of the cell type specificity, which one can construct and input that as histogram as part of the model averaging, which we thought we should do, but we did not do it. So that's one more information which is there in the paper and its data. So as such, we only know it's somewhere there. So, or you could use that histogram from that cell paper to model average over all values of K and weight it according to what has been observed in that paper. Also, that might be less ad hoc than putting K equals 4, let's say.
And a final question is, do you have any other applications in mind for your algorithm, or was this just a fun challenge to do, or both?
It was just a fun challenge. I'm a physicist, so I'm not going to use it. Perhaps Chaitali can use for her metastasis projects. I don't know. You want to take it?
I mean, we basically did it as a fun project, but I mean, given now we have developed this, I mean, we're thinking if we can apply in more disease-related conditions, for example, looking at Alzheimer's, or even since I'm a cancer biologist, if looking at, metastasis or the brain tumor, how the landscape of the brain changes. I mean, if we can use these kind of approaches would be good. But to begin with, it was just a fun challenge. We were just, interested in doing it.
All right. Well, thank you everyone for presenting in today's webinar. We really appreciated all the effort that went into submitting, your submissions to the Data Challenge and also putting together these presentations. And again, this will be available to watch again, or for anyone else to watch on our YouTube channel in about a week or so. All right. Thank you, everyone.
All right. Thank you.